these? Those are Z-discs. And what is it from Z-disc to Z-disc? A sarcomere. So this is the contractile unit of a muscle, is this little thing from Z-disc to Z-disc. That's our sarcomere. That's what does the contracting. So within a sarcomere you have different myofibers. What are the two myofibers? Actin and?
That's a thick and thin.
Thick and thin also works.
Yeah,
So the thick fibers are made out of myosin. The thin fibers are made out of actin. So here are thick fibers represented here. They have the myosin heads on them. Here are the thin fibers, and we'll talk about all the components in there in a minute. Okay, so from here to here. So it is the Iban. How do you know it's the Iban? So for those of you who can't see, it's like from there to there. It's just the thin filaments, it's just the actin filaments. That makes it the Iban. Can you tell why it would be, and is it light or dark? How can you tell it's light just looking at this? Because when it's just the light, when it's just these, the thin fibers, you don't have those thick fibers in there, so it actually reflects back as a lighter band. That makes sense?
Yeah.
Right, so from here to here, it's light. So what about from here to here? If you guys need to move, feel free. This is the A band. Is the A band light or dark?
Dark.
Dark. Does it make sense why it's dark? Because you have overlap of the thick filaments with the thin filaments, so it makes it reflect darker. Have I lost anybody yet? Okay. So within the A band, this is where it's within the A band, there's actually a lighter dark band. And that's between here and here. This is the H zone. Why is it a little bit less dark?
Because
There's no overlapping actin. So the H zone is a little less dark. In the middle of the H zone, you have a darker zone. Your M line. The reason it's darker is because you have something called endomysium. They're proteins that hold your myosin filaments in place. They tether them. So that's what makes this little M line so dark, and that's what the M line actually is. That make sense? Don't worry, I don't think she wants you to know that. I was just letting you know there's a protein in there. It tethers the myosin filaments. So we also have these. What are these little green ones? These are the troponin complex. The troponin complex hold this yellow one in place. What's this yellow one? The tropomyosin, right? So the yellow is the tropomyosin. So you have these troponin complexes. It holds the tropomyosin in place. What are these little red spheres? Those are the actin molecules, specifically G-actin. It's actually a type of actin called G-actin. These actin actually have what's called an active site. And an active site is a place where we're going to have something bind. What do you think is going to bind to those active sites? The myosin heads. So the myosin heads are actually going to bind to the active sites on the G-actin molecules. The problem is the tropomyosin is actually covering them. Right? And what controls the tropomyosin? Troponin complex, ultimately calcium. So the troponin complex will get activated only in the presence of calcium. That makes sense so far? So what happens is, if these active sites are open, the troponin heads bind and they ratchet forward, like this kind of motion. That's going to do what? It's going to pull the Z line closer to the M line. That is our contraction. So when those sites are open, the myosin heads, you guys talked about them being in the cocked position, right? They're ready to go, and they can start ratcheting. They do what's called a power stroke. First of all, what is this? It is a muscle fiber. All right, so here you have your bone, then you have your tendon, right? As you get closer and closer to your muscle, think of your bicep for this example, it's just easier. Around it you have what type of tissue? What do we call it? The tissue that surrounds the whole muscle. It's the most outer layer, right? Your epimysium is the most outer layer of the muscle. Fair enough? So it covers the whole muscle. If you look inside, you're going to see all these little sort of, can you guys see it from here? They look like little individually wrapped components. Each one of those is a fascicle. So they pulled out one of the fascicles here, which is what you're looking at. What is a fascicle wrapped by? That's perimysium. So within all of these fascicles are hundreds and hundreds of muscle fibers. The equivalent of like a muscle cell, but we call it a muscle fiber. That is a muscle fiber. So what they've done is they've pulled out one of the muscle fibers. What surrounds a muscle fiber? I'm so sorry. Endomysium. And then what if I pulled out one of these from the, what if I pulled out one of these little circles? That would be a myofibril. So technically, each one of these little circles that you see in this model are little myofibrils. You know it's that area because you can see how it's like the alternating dark light, dark light. You'd only be able to see that here. Does that help? Does that make sense on the layers now? So we're specifically looking at a muscle fiber. So what does that make this outermost layer? The endomysium. What is this? Just deep to that, we have this and this. That is the sarcolemma. Right? So the sarcolemma is, and what is the sarcolemma? It's a modified plasma membrane specifically in muscles. Right? And so that's what you're seeing here, and it travels up the walls. That is your sarcolemma. You're going to notice, if you look at it this way, that the sarcolemma starts invaginating into the muscle, right? You guys see that? Where it starts coming in, it starts invaginating, so it's nice and smooth, and all of a sudden it starts doing that. That is where the nervous system meets the muscular system. So I know you guys talked a little bit about this. I'm not going to talk about the specifics of the channels. If you want me to do that, I can do that maybe next week or later. But the nervous system is going to send an impulse in. It's going to release acetylcholine into this area. This would be a synapse, but it's a very specialized one. We call it the synaptic cleft, but this is specifically the neuromuscular junction, right? This coming together of the nervous system and the muscular system. We release a bunch of acetylcholine into there, in that synaptic cleft. There's a bunch of acetylcholine receptors on the other side. You get depolarization, and you start sending a signal down the sarcolemma. Everybody with me so far?
So is that the depressions in there, would that kind of be motor and plate?
Yeah, those are, the motor end plate is going to be that area where it meets, yes. Mm-hmm. And so you have this signal that's traveling down the sarcolemma. And what else is continuous with the sarcolemma? What other structure? The T-tubules. Which ones are the T-tubules on here? The blue one. You can actually see how on the sarcolemma here you can see these little holes and they're meeting up with these blue T-tubules, right? These are all, that's how it connects. And so that propagating signal can actually travel down these T-tubules and right flush up against the T-tubules are what on both sides? The terminal cistern. What is a cistern in English? Like a tank. Like a tank. So essentially think of them as just a tank full of calcium. These are just tanks full of calcium. So when the signal comes down, you open up special channels, special voltage gated channels. And those voltage gated channels are going to release calcium from the terminal cistern. Why is that important? Who cares?
The troponin and the whole like binding of the myosin.
It's because of the troponin complex, not the tropomyosin, but the troponin complex. So all of this nonsense, all of the anatomy, all of the other physiology that we're talking about here is to do one thing. It's to release calcium from those so that we can bind the calcium. You guys remember the little troponin complexes? When you bind calcium to that, what's going to happen?
They're gonna open.
They're going to be uncovered by the tropomyosin. The troponin complex is going to move the tropomyosin out of the way. Your active sites are going to be revealed. The myosin heads can now bind, and you can get your power stroke. And then we get our contraction. Does that make sense? So you're going to talk more about this process. If you want me to go over some of the other specifics, I can, but it'll probably be next week maybe. So another question for you guys. What happens to your H zone during contraction? It disappears. Why? Yeah, and how did we define the H zone? It was just the myosin fibers. When it contracts, notice the actin fibers actually cover it now. We don't have an H zone anymore. Well, I can't make it go all the way together. But your H zone would actually disappear. Make sense? Yeah, it's just when it contracts, right, everything comes together. And then there's no more H zone anymore. Because then the actin and the myosin are everywhere. So it's just one big dark band.
And then, so there's no more.
Make sense? Other questions?
Merry Christmas.